Read, I saw your office, you have more than 10 of those books for publications. This is only one of the example, but Dr. Morre published over 750, papers, mostly about this inox-2, about the inox protein. I am very fascinated about it, and I want to know what's the story behind it. How come you devote almost half a century your life to study this, to the cancer research? Tell me about your story.
Well, it started very early. I think as a youth, in high school, I developed an intense interest in how plants grew. When I graduated from high school, my first job was at my local university, at the University of Missouri, and I was hired to conduct research on herbicides, chemicals that basically kill plants.
All right.
And my very first assignment, my very first job at the University of Missouri was to spray some weeds that were growing in a fence at the university farm. And it was right along the road at the driveway into the farm. And so every morning when I went to work, I would go by these weeds, and at night, going home. And so I observed them every day. And 2,4-D, which is a herbicide, kills plants by causing unregulated growth.
Okay.
And these plants would grow all directions, they would swell, and they did this for several months until they finally died. And this unregulated growth reminded me of cancer. And I realized, actually at age 16 years old, that if I could understand how 2,4-D killed plants, I could understand how cancer killed people. And I was correct.
Wow.
I worked on it the rest of my life.
Wow, you must have, great drive and great courage to stay in this,
absolutely,
in this
absolutely.
path.
Because it was completely out of the box. I think nobody would have ever recommended that I follow that route.
In this process, so many years, I know you must have had difficulties, misunderstanding, those very difficult, because I have not known anybody else doing this through and stick on with it for so long time. And tell me about the difficulties you encountered and how did you overcome it?
Well, let me just say a little bit more. As I just said, the beginning in cancer research was really the summer of 1953, when I was 16 going on 17 years of age. And at that time I really decided as a career goal to study plant growth rather than to pursue my original plan was to become a veterinarian, to study veterinary medicine.
And, you think people more important than
And my undergraduate advisor, I spoke to him about what I wanted to do, and he recommended that I, instead of just taking plant courses, I should study basic sciences and chemistry and physics and math in order to pursue this career. And that was probably the best advice that I was ever given in my whole life. So I did. I followed in a very basic course of study. And for the next 6 to 8 years, I followed this idea, trying to understand how 2,4-D killed plants. I got my PhD from Caltech, from California Institute of Technology in 1963, and accepted a position as an assistant professor in Lafayette, Indiana, where we are now, at Purdue University. And I always kept at this one problem. People, they were amused, but they said, what is this guy doing? Because at no time in my entire career did I ever simply repeat someone else's experiment.
Most of the people do that way.
Yeah, all my research and publications all of them are original works, and there's no precedent in the literature. My vision was that of a common denominator, something that was common to all forms of cancer, which ran totally against conventional wisdom at the time, and it would be something that was also shared by plants, and something that could not be explained by genetics. Now, the biggest problem that I had, you asked about that, was to uncover what was that common denominator. what was there that all cancers had, and was also found in plants, and that led to cells growing out of control. The solution came in almost 30 years later, by the discovery published in 1986 of a growth-related protein. It was an NADH oxidase on the surface of the cell and ecto on the cell surface, and we refer to that now as enox. It was induced by the herbicide 2,4-D in plants, and was induced in cancer, and was directly responsible for the uncontrolled growth that led to unregulated growth and life-threatening conditions in both the plant treated with the herbicide and in cancer.
So there was a close relationship between
very close relationship.
Yes, and
they're both enox proteins.
They are, even in the plants as well. Wow. Okay.
Now these initial observations from plants were, and we quickly then confirmed them in liver cancer in rat liver. We had rat liver model, and we discovered that the responsible proteins in both the rat liver and the plants were unique proteins. One in plants was induced by 2,4-D, the one in rats and later in humans was induced by cancer, and both led to unregulated growth and, as I said, a life-threatening condition because the plants died and the rats would die. Now, the main disagreement with our findings, and part of the trouble that they're still not universally accepted, is that there's a general premise that's widely accepted that all cancers are different.
Yes.
Lung cancer is different from breast cancer is different from prostate cancer, et cetera, et cetera, et cetera.
Yes.
And that a common protein absent from normal cells and tissues and shed into the blood serum as the cause of unregulated growth in all known human cancers is impossible.
Most people think impossible.
Impossible.
But you
can't
find
be.
it is possible, and it is a real thing.
And even in the face of very, very, very compelling evidence, they still say there's something wrong. Your experiments are flawed. Your hypothesis is flawed. It can't be. There's no such thing as a universal cancer marker.
But you use the science, you use the molecular science, you use electronic microscope, and you did all the DNA sequence, and you really found the solid theory and solid test and to prove it.
Absolutely, absolutely. We have a 570-some page book, nothing but the evidence, all peer-reviewed.
I'm very impressed. I'm very impressed. I have that book I bought and give many people. I read many times, but every time I read, I still get new from that.
The oncoblast test, which developed out of this, and the enox-2 protein.
Yes.
There's no smoke and mirrors here. It's real. It's very solid.
Because you purified it,
everything,
and you have the microscopy and took the picture of it, and you have the DNA sequence, and you really
RNA sequence, protein sequence.
You got the down to the real
and we have
molecular level.
And we have proof that it is, first of all, necessary. Enox-2 is necessary for a cell to become cancerous and have unregulated growth, and it's sufficient. No mutations are required. All that's required for a normal cell to become cancerous is to express Enox-2. So it's both necessary and sufficient.
I want to show you, this is Dr. Marie's book. It's over 500 pages, and also the reference is 2,500 papers.
And that's published together with my coworker, a longtime coworker and wife, Dorothy Morre. So she's a co-author on the book.
Yes, yeah.
And also an author on most of my papers, and we've shared students and ideas and hardships throughout the whole years. And she's also a developer, co-developer of ankle blood test.
Yes, I will get to interview her, a short time later. And also in those, papers, you yourself published, I think, over 750 papers, and those papers are the most mentioned by other scientists. They refer your paper, which I really think you draw a big attention, people. But let's get back to the question about, why it is necessary for the cancer INOX2. Give us some of your science.
Just to maybe point out some of the specific experiments that were done. I'm looking at here a 2004 publication. The senior author is Pinju Chu, who was a graduate student of Mrs. Dr. Morre for many years, and she got her PhD with us, and is now a professor in Taiwan. And what we did in this was to use non-cancer cells,
okay,
and transfect them with the INOX2 gene,
okay,
and look at the response. We have different kinds of experiments that we did, and one is simply overexpression. We would have normal cells which grew at a normal rate, and then we would overexpress the INOX2 in those normal cells,
okay,
and then they would grow like cancer cells.
yes,
They would divide in an uncontrolled fashion and very rapidly. There are some differences between the INOX2 and the constitutive INOX1 is present in normal cells. The activity has a distinct periodicity. The period length is 24 minutes for normal cells, 22 minutes for cancer cells, and the INOX2 is inhibited by a large number of cancer drugs like adriamycin, cisplatin, and so forth. And so the NOx activity in the normal cells had the 24-minute period and was refractory to the drugs, was not affected. But as soon as the INOX2 is expressed through transfection, then the period was shortened to the 22-minute period, and now it was inhibited along with the growth of the cells by these drugs. Another, and these are just more growth experiments and so
let
forth.
me understand, in the layman's word, it sounds like, you get a normal cell,
Yeah.
and then they do not express
INOX2.
INOX2, and then you transform those cells into cancer cell.
Yeah, by adding only a single protein, and that's a single gene, a single piece of DNA, and that's the DNA to make INOX2.
Okay, and then they become cancer cell.
Yeah.
And then that time you measure, it has INOX2
Right.
protein. So that is the theory, or that is your evidence
Right.
to show us, on the normal cell there is no INOX2, and then when you put into the gene for producing the INOX2,
Yeah.
and then it becomes cancer cell. And this time in the cancer cell you measured it has INOX2 protein. Is that correct?
Right.
That's how you can tell. This is the evidence. INOX2 only present in the cancer cell, not in the normal cell. Is that the case?
Yes, thank you. Yes, and here's a little table of data where we show that the INOX2 transfected cells are inhibited by six of the most common anti-cancer agents, drugs.
Okay.
Whereas the normal cell before that was unaffected at the same concentration. This is a very interesting experiment here. This is looking at the invasive ability of cells by growth in matrigel. Matrigel is a matrix with pores in it, and normal cells won't enter it.
Okay.
But if we and they just stay on the outside and don't do anything. But if we put Enox-2 transfected cells, that the transfected cells will move right through the matrigel, and it's a model for metastasis. Because the hallmark of cancer is the ability to invade.
Right.
And normal cells will not invade until they have Enox-2, and as soon as they get Enox-2 gene, then they can invade.
All right. So then that's another evidence you can see. Those are the cancer cells because they can invade, they can cross border, they go to other tissue.
And there are just two more bits of evidence in this necessary and sufficiency argument. One is that the classic test for cancer cells is their ability to grow on soft agar.
Okay.
All right?
Yes.
And so if you take normal cells, they won't grow on soft agar.
All right.
And if you transfect them, then they will grow on soft agar just like cancer cells.
All right.
And if you take cancer cells and treat them with antisense to knock out the Enox-2, then they can no longer grow on soft agar. They develop a normal phenotype.
Wow.
And vice versa.
Okay.
And then one of the major early developments of cancer biology was the discovery of the SARC gene, where was a virus, a chicken virus, that would cause cancer in chickens.
Mm-hmm.
And it was one gene in this virus that was responsible for that. So this was the first evidence that that there was a tie to genetics.
Mm-hmm.
And this was followed not too long after that by the discovery of the first human cancer gene, which was RAS.
Uh-huh, right.
And we've also done studies with RAS.
Mm-hmm.
And cells transfected with RAS make Enox-2.
Wow. So.
And if they're not transfected with RAS, or non-transfected cells don't. And then we have a model for the viral transformation.
Okay.
SV40, which is a transforming virus from monkey virus in monkeys,
Right.
but will transform human cells.
Mm-hmm.
If we take normal cells like umbilical cord cells,
Mm-hmm.
and transfect them with SV40,
Mm-hmm.
then they will make Enox-2.
Oh, right.
So
then they become cancer cells.
Yeah, and so the viral origin of cancer involves Enox-2, as does the genetic origin of cancer from the RAS gene and related oncogenes.
Wow.
So the common denominator to cancer is Enox-2. It's both necessary and sufficient.
How many years it took you to find out this? And I understand it must be taking long time and numerous experiments and significant amount of your life, your energy to putting into this, especially in the high pressure and nobody else believes on it.
86 to 96, 96 to 106 is 10, and 26, and 2001 was that? That's roughly 20 years. 20
years, just to prove, this Enox-2, it is necessary and it is sufficient for, the cancer cell to spread and to be a cancer cell. That's correct, right?
Yeah, a little over 20 years, 25 years.
Wow.
So the initial discovery was in 1986 of the NOx proteins, and the necessary and sufficient paper was published in 2004.
This whole picture, gene mutation, DNA, fragment mutation, and to mRNA transcript, and to the protein production. And most people think the cancer is a gene mutation. We believe some of them true. It's a gene mutation and then it leads to cancer. But I understand you do not completely believe that, because you think when the gene mutation and then there is a chance to repair, and then maybe recover. But to the level to the mRNA part, the level, and then down to the protein level to express ENOX2. And as you told me, you said ENOX2 gene is in normal people. Give me a little bit more explanation and the whole picture about the whole process.
Okay, yeah, great, thanks, Dr. Wu. That's an excellent question. No, ENOX2 is not the result of a mutation. The ENOX2 gene is present in everyone.
And that is in the genomic bank?
Yeah.
You have the whole sequence.
Sequence and everything like that. The messenger RNA is produced, but it's not translated into protein.
Okay.
So
This is
everybody
for
has the ENOX gene. They make ENOX2 messenger RNA, but it's not translated into protein.
Okay.
And it's very clear that the full-length 71 kD ENOX2 protein is simply not translated. And we've got probably a good half a chapter in our book explaining how this works. And what we initially found was that cancer cells have splice variants of ENOX2.
Okay.
And the common splice variant that's found in all cancer cells is an exon 4 minus. ENOX2 has a number of exons, total of 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13 exons, and one of the exons is removed. Now the protein, a new messenger RNA is made, and that protein is expressed at the plasma membrane.
Okay.
And if we do an exon 5 minus, that doesn't work. And the result of the exon 4 minus expression is a 34 kilodalton fully processed ENOX, which is delivered to the plasma membrane. So it goes, it's translated, it shows up in the endoplasmic reticulum, it's transported to the Golgi apparatus, and then processed, shortened, molecular reduction in molecular weight, and delivered to the plasma membrane. The new messenger RNA is initiated at methionine 231. If methionine 231 is blocked, then exon 4 minus splice variant is also blocked. And we've done subcellular localizations and all this sort of thing, and only with the ENOX 4 minus protein it does the splice variant, does the protein get delivered to the plasma membrane as functional ENOX2. So it's not a mutation.
Okay.
It's a splicing problem. And we are working on that, and we've recently reported that the splicing factor HNRNPF directs the formation of the ENOX 4 minus variant, and we're looking at the spliceosome complex, isolating it, and to look at what protein now signals the formation of this spliceosome complex and the actual splicing, appropriate splicing process, and that it probably has something to do with some sort of developmental stress or something. We're unsure exactly what it is that triggers it, but the known oncogenes, for example, whatever they do is what causes this splicing event to occur.
Okay.
And so we're looking at SV40 virus transfected HUVEC cells and human umbilical cord cells, and to see the protein changes, what complexes with HN or NPF to result in this all important exon 4 minus.
That is one of the experiment you're doing now. I know in your lab you have, there's many people doing so many different projects and, like really high-tech, science behind those walls in your lab. And so that is one of the things you're doing. As I understand, it sounds like, DNA promoter and the DNA damage is more upstream, and then down to here, like a unhealthy lifestyle is more on here. But cancer is a chronic disease. It's taking 10 years, maybe 20 years to really develop to full-blown cancer. So those are in the early stage, but down to mRNA to this. So sounds like every different cancer, no matter which form of cancer, which type of cancer, all have to go through the last step, which is the INOX2 presence in the cell membrane, outer cell membrane, and to produce INOX2 is necessary and is a must for cell become cancer and to
without
spread.
the INOX2 protein in the plasma membrane, the unregulated growth simply will not take place, and the invasive ability will not be acquired.
So looks like this is a downstream. It's the last step.
So last step, right?
And then it is that is why testing this. It is the.
And there are many paths. There are many paths to the induction of ENOX2. But the cell will not be a cancer cell until the ENOX2 is induced.
All right.
You can induce proliferation. You can have cells that are precancerous in response to chemical carcinogens or some other environmental factors, and then at some point this splice variant will be produced, and then one is faced with metastatic disease and clinically defined cancer.
Understand. So sounds like we know the INOX2 is only happens in cancer cell. So would that be good measuring the INOX2 and the amount or whatever? Is that a good drug screening?
No, I think that since ENOX2 is probably the only molecule that's specific for cancer, that it's an excellent opportunity to screen for anti-cancer drugs. As it turns out, there are many effective anti-cancer drugs out there that do use inhibition of ENOX2 either as their entire basis for activity or at least partial basis for their activity.
And also it would be good for now nutraceutical. Right now nutraceutical is very hot and people are trying to use a natural way, like Chinese people using Chinese medicine for several thousand years, try to kill the cancer. Cure the cancer, and so that's how you come up with some nutraceutical and screening and to find out it does work or not work for the anti-cancer.
Yeah, the nutraceutical screening has been especially interesting. It's very clearly it's the target for the anti-cancer activity of green tea, for example.
I understand.
The anti-cancer activity of capsaicin is also targeted to ENOX2.
I understand you have that product and you published the paper about it. Right. Sounds like it's very effective. It could get to over 90% for early cancer patients.
Right.
That's congratulations. I'm thinking that must be a historical moment when you first find out this, the ENOX2 protein and how it relates to the cancer cell. And so describe a little bit, tell us about your emotion and what that day look like. How did you find it finally after so many years experiment?
Well, as I said earlier in the interview, the biggest problem was to uncover what was the common denominator between cancer and plants sprayed with herbicide. And I have to go back to that part of the research. And this is in the early mid-1980s. And I was serving at that time as a charge de coeur at the University of Geneva in the plant biology department, and I would, Mrs. Dr. Morre and I would go to Geneva occasionally, maybe once or twice a year, to lecture and work with students and do experiments. And at the time we knew that we were fairly sure that the common denominator protein was a redox protein. Probably has something to do with oxidation of NADH. It would be stimulated by 2,4-D in plants, and very little else. And one of our colleagues at the University of Geneva worked on peroxidases, and I was complaining. I said, we've looked at every electron acceptor that we can think of, and none of them are stimulated by 2,4-D. And he says, have you tried ascorbic acid? And I said, no. Or dehydroascorbic acid, no. And he said, well, when I work with peroxidases, we find it on the outside of the cell, so it might be the electron acceptor. Mm-hmm. And so I said, no, it's a great idea. So let's check it out. And we were sitting in a nice Swiss cafe in the center of Geneva, drinking Swiss white wine, and I don't remember exactly what we were eating, but it was delicious. So as soon as we finished our lunch, we went back across the street to the lab, and we had made some soybean plasma membranes that morning for another experiment. And I always carried 2,4-D with me wherever I went. And we found an old bottle of NADH. We found a spectrophotometer that would work, and we put it and some ascorbic acid. We put it all together, and for the first time, we got a strong stimulation by 2,4-D.
Wow!
So that was it. It was and then we realized after about a week that the ascorbic acid was not even necessary. That the acceptor was really oxygen. And so all you had to do is to have NADH and the enzyme and add the 2,4-D, and it would oxidize NADH. Mm-hmm. And all our experiments with these artificial acceptors was just masking the real protein.
Okay.
After that it was just all downhill. We knew what the enzyme was. It was NADH oxidase.
Wow.
You know, that was the turning point.
That's the moment you celebrate and you're happy.
Yeah,
I think that
Actually, it was in reverse order. We celebrated and then we did the experiment. I thought it was a capital idea. It was the only good idea.
So that nice cafe must add a good flavor for your body and for your mind.
I have pictures of that cafe, and I have pictures of the table.
For the memory.
Yeah, and the person who suggested the experiment is a co-author on the paper. So.
Wow. All right. Yeah. So after so many years, so many basic experiments, you found out, you are very much familiar and get to know this INOX2 inside and out. Now you developed a test called OncoBlot, I understand. Mm-hmm. Tell me about this OncoBlot. What you are imagining? What kind of test it is? What would you tell your people, and what kind of people should take it? Just give me some idea.
Well, the OncoBlot test grew out of our research with the INOX2 protein. We knew that the protein was on the cell surface, that it was shed into the serum, and that we could determine INOX2 presence in human subjects as an indication of cancer presence. And we considered developing a test, but there were some, I would say, some ethical concerns with this. Presumably we could detect cancer very early, maybe earlier than any other method. And the recipient of the test would have no information. This organ side of the cancer would be unknown, and there was nothing they could do about it. Because even today there is nothing, there's there's no standard treatment for early cancer detection or early cancer intervention prior to clinical symptoms. There isn't anything, because the reason there isn't anything because the only way you could use it is with some sort of very early detection method. At that time that didn't exist. So my wife and I formed a small company while we were still at the university to try to develop the test further. And one of the techniques that we employed in the new company was two-dimensional gel electrophoresis. We already had an ENOX2 specific antibody that we could use for Western blotting, but on gels of serum, the patterns were just too complicated. We couldn't really interpret them. Again, we could tell whether the person had cancer or not, but that was all we could determine. And I was at the time was working with with our oldest daughter, and she had connections. She was a compounding pharmacist, and she had connections to a cancer diagnostic lab in northern Indiana, and they gave us serum samples, and they just, at the end of the day, they just pour all the leftovers into a beaker, and we got this mixture with all different kinds of cancer in it. And so we ran a two-dimensional gel with this mixture, and we had spots all over the place. We had maybe 20 spots. And I'm the eternal optimist, and it's oh my goodness, everything's lighting up. Your antibody's no good. And I said, well, maybe that each one of these represents an individual cancer. So then we started running sera one at a time, breast cancer, lung cancer, and it was true that in the 2D system, each tissue of origin gives a specific characteristic pattern in terms of molecular weight, number of spots, and isoelectric point. So that we now have a database of in the thousands of patients, and we have 26 different cancers in our database, and they can all be distinguished one from another.
Wow.
And what apparently happens is that each tissue of origin constructs a slightly different splice variant. The splice variant that's expressed is slightly different.
Okay.
Each splice variant contains a common sequence, which is the antibody combining site and the drug binding site of the ENOX2 protein. So that particular exon is present, but there can be additional exons or less exons, and that is tissue specific. So we developed this as the Oncoblot tissue of origin cancer test. So now we had a really great system where not only could we detect cancer early, but we could identify the organ site, so people could maybe do some follow-up tests to confirm the cancer. And then we did a fairly extensive clinical trial, which we published last year with patients who did not have cancer or a clinical history of cancer, just people who weren't expressing clinical symptoms, and those that presented with a spot in Oncoblot. These were given the nutraceutical that Dr. Ping was talking about, which is directed to the ENOX2 protein, and for 3 to 6 months or so. And then we went back and retested them at the end of their their nutraceutical intervention, and 94% of them, the early indications of cancer were gone.
Wow, that's
And
great.
in the 1 or 2 years since that trial, none of these people have yet to develop cancer.
Wow.
There was in the trial there was one person who elected not. He tested positive for lung cancer. He elected not to take the intervention therapy, and was clinically diagnosed with non-small lung cancer 36 months later.
36 months later.
Yeah. And there was another subject who had an oncoblot tissue origin test indicating leukemia or lymphoma. We can't really tell them apart. A blood cell cancer. Withdrew from the study. Elected not to take the capsule T, and was clinically diagnosed with lymphoma 10 months later.
Oh, okay. Okay.
Whereas in the study, it's been 2 or 3 years now, and nobody has come up with cancer yet. So we're encouraged by this.
Again, how many months now those people are after taking the capsule T and still free of oncoblot still negative? How many months? Most of them?
Well, it varies, but it's order of 2 to 3 years.
Okay. Okay. Great. Great.
The importance of early detection, Dr. Wu, I think is something that everybody that's associated with cancer can agree on.
So it's true.
That is the key. That's going to be the key to the conquest of cancer.
We
With
all
early
know.
detection, I think you have the possibility of converting or of taking a disease which is now an acute disease. Stage 4 is basically incurable. I think I agree with that. Almost everybody agrees that the possibility of recovery becomes less and less and less as the cancer advances. But I think with early detection, early intervention, we basically make it a chronic disease, something that's manageable.
Right, yes.
And
right.
and I think everybody agrees to that. What is missing from our arsenal are therapeutics directed at early cancer. Because all the therapeutics that we have, the chemo, the radiation, and everything, this is a very harsh treatment, and nobody would think about using these on a very early cancer where there may be only less than 1 million cells involved. So we need to rethink our whole concept of intervention.
Yes.
And come up with early interventions that are completely safe and effective against small numbers of cancer cells in the body.
This oncoblot, based on your over 40 years research, is really solid science based on, and now I think it's quite interesting, it's quite useful for test early cancer. And as you said, when you test this, you detect the INOX2 protein present in the patient's serum. You can predict this patient have cancer cell. And so then we can take on the early detection and take on early action about it, as you said, nutraceutical, and which benefit the patients much more, which is a great thing you give to the society, give to the human being.
I think it opens up a number of avenues which have been explored certainly, but never brought into a clinical practice because of their ineffectiveness against late stage disease. I think for the first time we really have a very good target for a cancer vaccine. I think it's a new opportunity to relook at immunotherapy, at immune system, lymphocyte activation, and this sort of thing. But I think the main thing is to look at basically start at the beginning of drug intervention, looking not at drugs that are so powerful that they will affect stage 4 cancer cells, but much milder, perhaps drugs that were tested and the the compounds from pawpaw, the annonaceous compounds, for example, so these sorts of things that were maybe abandoned because of their inability to cope with stage. They might be very effective against early cancer.
Wonderful, wonderful.
Yeah.
Sounds like this test is pretty easy to do. You only need to take a 10 cc of your blood and then spin down as a serum, and then you run the 2D gel. You can detect different amount of this, and then you also can detect around 27 origins. So that sounds like a great tool for in the detection of the cancer. It's a great tool. And as you said, later on you may develop cancer vaccination, and you also can do drug screening, and to see the patient who already have cancer, whether they have recurrent or not. For the patient who have strong family history, they need to do the test because it's an easy test to do. So you can get them on early detection. It's all very good. Good.
Yeah, well, early detection, the opportunity for early detection is definitely a paradigm shifting event in the conquest of cancer, and I'd be very pleased to continue to be a small part of that shift, that paradigm shift. So,
wonderful,
thank
wonderful.
You very much, Dr.
Thank you, thank you, thank you. Admire you, both of you. And as I understand, you are the founding director for Purdue University Cancer Research Center. Do you remember which year was that? Yeah,
that became founding director of the Purdue University Cancer Center in 1976, and I held that position until 1986.
Wow,
and
10 years.
During that period, this was near the beginning of the National Cancer Act, and we received a center grant from the National Cancer Institute to form our center, and we provided new faculty in different departments, and this led to ultimately to the construction of a cancer research building.
Okay.
We got a construction grant from the NCI, and I was able to move into the new building, and I was the first lab to occupy the new building as it was being constructed, and I had my lab in there. I was all by myself in this good big building. Construction workers all around. It was fun. All right.
Wonderful, wonderful. And now we turn to Dr. Dorothy Marie. Dr. Dorothy Marie, as I understand, you are behind your husband, Dr. Jim Marie, for so many years. Both of you have been working on this INOX2 protein for so many years. What department were you from? And what I understand you are also on biochemistry. You taught biochemistry courses. Tell me a little bit about yourself.
Okay, I was in the Department of Nutrition. I did teach a course there in metabolism for many, many years, and it was biochemistry oriented. So that was exciting for me because I would go home in the evening or so, and we'd talk about biochemistry and some of the reactions and so forth, and our children would say, "Mom and Dad, are you still doing
work?" So and work is work, go home is still work. Yeah.
But we borrow each other's transparency, slides, and PowerPoints, and this sort of thing. It was very, very, very synergistic.
Yeah, it was fun. Really.
We should have written a biochemistry text together. It just wasn't time.
Well, I was going to with a lady, and then she decided she didn't really want to put that much work into it.
But you are the person behind this book, behind all this research, every step behind, on this. So we also very admire about you, and we are very proud of both of you for doing this.
It was quite an experience to write the book. We worked together very much. Of course, we had other people helping us as well, and it was fun to put it all together.
And I would say that just about at least 50% of the INOX2 and INOX1 work
Mm.
that went into this
It
book
was-
was from her laboratory,
Oh.
and roughly half of mine. And we shared students and have a lot of joint publications.
Wonderful.
And people would ask me if I was working in his lab, and I said, no, we have a street between our labs. And that was important.
That's wonderful, wonderful. Yeah.